I celebrate and acknowledge the traditional owners and custodians on the land on which we meet today, the Wurundjeri people of the Kulin Nation. I pay my respects to the elders, past and present, and all Aboriginal and Torres Strait Islander peoples from other communities who may be here today. And so I'll introduce Thomas. Thomas is a cancer cell biologist working in the field of the extracellular matrix and ECM remodeling in the progression and metastasis of solid tumours. He graduated with a PhD from the University of Durham, UK in 2008, followed by postdoctoral research positions at the Institute of Cancer Research in London, UK, and Copenhagen University, Denmark. In 2016, Thomas was recruited to the Garvan, where he currently leads the Matrix and Metastasis Lab. The lab's creative research program integrates matrix biology with precision oncology, aligning with the Garvan's strategic vision to make fundamental advances in the understanding, treatment, and prevention of human disease, in this case, personalized stromal targeting in solid tumors. Thomas takes an active role in training emerging scientists, honours and PhD students in their projects. And Thomas is the president of the Matrix Biology Society of Australia and New Zealand, as well as Director of Communications and an executive board member of the International Metastasis Research Society. And Thomas is also a co-founder of the Australian Pancreatic Cancer Matrix Atlas. So thank you very much for being here, Thomas, and yeah, please take it away.
Great, thanks Somali for the introduction, and also just thank you to the organisers for giving me this opportunity to come and talk to you today, and to give you some insight into the tumour microenvironment. So as mentioned, I'm based at the Garvan Institute here in Sydney, and have been now for about 5.5 years or so. And so my lab is interested in really how the extracellular matrix, which is sort of a key component of that tumour microenvironment, really feeds into the initiation, development, progression of solid tumours, and also how it affects response to therapy. And so as my sort of, I guess, remit for today is to really go through with you guys what is the tumour microenvironment, what are some of the most important components of the tumour microenvironment, and given the fact that most of my research is based around the extracellular matrix, what it is and also what role that plays in the tumour microenvironment. I'm going to talk a little bit about some of the ways which we've been developing to study the extracellular matrix in tumours, and then touch on why the extracellular matrix is important in cancer research and also treatment. And if we get time at the end, what I'm going to do is run quickly through a research example of one of our recent publications looking at cancer cell matrix interactions in pancreatic cancer. So what is that tumour microenvironment and what is it made up of? Well, I'm sure you're aware that tumours are complex ecosystems. They are made up of more than just cancer cells. But what's important is it's not just cells, that there are acellular components of a tumour as well. And collectively, all these things together are known as the tumour microenvironment. And so what I've just shown you on the right-hand side here is sort of a stylized diagram of a tumour. Now, you don't need to worry too much about some of the boxes around, but what I'm using this is an example to highlight how you have tumour cells. In this case, these are pancreatic ductal adenocarcinoma, or pancreatic cancer cells shown in the middle. But these are essentially surrounded by fibroblasts, immune cells of many different flavours, B cells, T cells, macrophages, and so on. So these are all depicted here. What's known as extracellular matrix, these are these little grey lines that you'll see around. You have presence of blood vessels within tumours, and an important factor as well is nutrients and other factors such as growth factors like TGF-beta shown here, but also things such as hypoxia. And collectively, that is considered to be the tumour. Now, whilst the malignant part of that tumour is the cancer cells themselves, they typically are the ones with the genetic mutations that are driving them to be malignant. The whole entity itself should be considered a tumour. And so a tumour doesn't just encapsulate these sort of central regions shown here of cancer cells, but actually extends much larger than that. And that's something I'll touch on a little bit later, is that actually in some tumours, cancer cells may make up less than 50% of the mass of a particular tumour. So my lab is interested in studying the extracellular matrix component of that tumour microenvironment. So what is the extracellular matrix? And for those of you who are aware, indulge me a little whilst I explain to those who perhaps haven't come across. So we know that cells are that basic building block of life, but how they come together to form tissues and ultimately organs is through interacting with and essentially with the extracellular matrix. And this is depicted here in the center. A lovely green GFP labeled cell here, and you'll see these sort of fibers. Now, I'm illustrating on the right-hand side here is with this video. So this, just to orientate you, this is a intravital imaging. So this is second harmonic generation imaging inside a mouse. The purple strands that you can see are collagen 1 fibers, collagen being the major matrix component within our bodies. The green are actually neutrophils that have been labeled with a green fluorescent protein. So this itself is not a cancer, but I'm using this video to emphasize a couple of points. The first is that if you watch these neutrophils as they move along these fibers, you'll see that their migration is essentially controlled by the presence of the fibers. They move along the fibers, they squeeze between the fibers. So we have a direction of communication in which the matrix is altering cell behavior. But if you continue to watch the movie, you'll see that some of these green cells are essentially altering those purple strands, those collagen 1 fibers. And here you'll see them moving them aside, in some places degrading them. And so this speaks to a concept we call dynamic reciprocity, the idea that the extracellular matrix influences cell behavior, but that cells can also change that matrix, which may go on to have further effects on cell behavior. And that idea of dynamic reciprocity is something I'll touch on a couple of times, and also something to keep in mind. So we talk about many solid tumours being what's known as highly stromal, stromal being another way of saying highly fibrotic, desmoplastic, or essentially just containing a lot of extracellular matrix and non-tumour parts, or non-tumour cells, I should say. And so what I'm showing on the left-hand side here, this is a H&E section. So this is a histology section stained with a dye called picroceres red. This binds to collagens and essentially using a very clever trick of the light known as polarizing light microscopy, you can look at the birefringent signal that's generated when this dye binds to collagen. And so what this essentially allows you to do is to look at the fibrillar organization and bundling of collagen. Now, the reason I'm showing you this is to highlight how you can see this sort of pseudo green and yellow sort of pseudo fluorescent image to show you what this extracellular matrix looks like around this tumor. This is a pancreatic tumor. And you can see that in this case, we've got this very dense fibrotic capsule forming around the outside that also appears to penetrate into the center of the tumor. However, it's not just this matrix does not just form at the edge of tumors. Using this example on the right-hand side of breast cancer, also stained with picroceres red, we can see that actually the matrix also penetrates deeply within tumors. And so we have both this extra or sort of invasive edge capsule, but we often also see a lot of matrix within these tumors. And this is important because in some cases, in some cancers, and pancreatic cancer is a good example, up to 50% of the mass of the tumor can be this extracellular matrix. And so it really just shows that we shouldn't just be thinking about cancer cells in isolation when we study tumors. Now, I'm sure most of you are aware of the different stages of cancer progression and metastasis, and I just want to use this slide really to emphasize one point, which is that the matrix and the tumor microenvironment as a whole really plays a key role, not just at primary tumors, where we might have invasion into or away from tumors and through interstitial tissues towards blood vessels, but also in secondary sites, metastatic sites, where we know that the matrix and the tumor microenvironment as a whole is important in the colonization of secondary tissues and also in dormancy, which is a very important feature of some solid tumors. But at all of these stages, it's this sort of cell intrinsic properties of the tumor cells, so the mutations which these tumor cells have, and that interactions with the cell extrinsic properties of the tumor microenvironment that really control progression. So when we think about the stroma, the matrix, the tumor microenvironment in solid tumors, I just want to make the point is that every tissue in our body contains a matrix, either soluble or insoluble. And I say that because if we think about the blood, even as a liquid, it contains a huge amount of soluble matrix components, which the minute you cut yourself begin clotting. And that's exactly what it's meant to do. And so this matrix is critical for the correct tissue function. But what happens is it gets dramatically changed in cancer. And just on the right-hand side, you'll be able to see some of the different characteristics of the matrix that are important. Composition in terms of biochemistry, binding of growth factors, hydration, cross-linking and stiffness, which I'll touch on shortly, which obviously in the biochemical properties, things like density, the actual ultrastructure, even degradation and turnover and the availability of interaction ligands, all play critical roles in normal tissues and are typically disrupted in cancer. And so again, it goes back to this point of this dynamic reciprocity between cell intrinsic and cell extrinsic properties. And just to highlight this a little bit, what I'm going to show you here on the right-hand side. So this is collagen 1. This is imaged via second harmonic generation imaging. This is a multiphoton microscopy approach that allows you to image collagen 1 fibers without the need to stain them. And what you can see, this is normal healthy fat pad. You see this rather sort of curly sort of collagen appearance. And on the right-hand side, you'll see the collagen 1 structure as tumors are growing. And so we can immediately see that there is a huge difference in the way that the collagen is organized between healthy tissue and a growing tumor. And so we know that these changes really sort of mark either transitional events in the progression of cancer or play a key role in regulating things such as response to therapy. However, what we also know is that there's sometimes similarity. And so what I'm showing in the bottom two images is this is collagen 1 in the normal lung. It's again got this sort of curly, wavy appearance. This is actually in part due to the ability of the lung to expand and contract during breathing. But in a developing lung metastasis, we see a reorganization of that collagen 1 structure. And if you look between the top right and bottom right images, you'll see that there is a huge similarity. And what this really says to us is that there are likely to be underlying remodeling events within the tumor microenvironment at both primary and secondary tumors that may be similar. And if we can understand both these similarities and differences, can we begin to think about therapies that might co-target these remodeling programs in order to improve outcome and treat both primary and metastatic disease? Now, for those who are interested in this, how this architecture and structure and three-dimensional organization changes, I just want to point you to a recent paper which just came out in Matrix Biology Plus by Raphael, Alejandro, and Janine. This talks about what we know as the matritecture, the matrix architecture. And so it's worth a read for those who are interested in understanding a little bit more about how that architecture is being studied, but also important in tumours. Now, something I just wanted to highlight and was that one of the biggest changes associated with remodeling of the matrix in the tumour microenvironment is changing stiffness of the tumour. And we all know, or at least anecdotally know, that tumours are stiffer than normal tissue. And many years ago now, some great work by Valerie Weaver's lab and colleagues showed that as transformation events occur and the matrix begins to be remodeled around a developing tumour, as you can see here, going from this curly appearance to these sort of straight linearized fibers, is that we see an increase in stiffness. This stiffness is known as, from a biomechanical perspective, to feed back onto many of the mechanotransduction pathways within the cell. But also, we look at it the other way and think, well, matrix remodeling has actually been exploited for diagnostics for many, many years now. As I mentioned, one of the first indicators of the developing tumour is often palpation and the feeling of a hard lump within tissue. And so that's really actually what elastograms, as this is a medial lateral oblique mammogram, and on the left is an elastogram, both are essentially highlighting or picking up changes in density and organization of the extracellular matrix as a result of the developing tumours. Now, that's not to say that it's only the matrix which contributes to these readouts, but the matrix and the tumour microenvironment is a significant indicator. So not only is this a way to diagnose tumours, but remembering back to the previous slide, it also subsequently activates many of the pathways known to make cancer cells more aggressive. So how does the tumour matrix drive disease progression? So we know that it alters cancer cell behaviour to make them more aggressive. I've mentioned mechanotransduction and activation of pathways, but also deposition of new or reorganized matrix can bind other cell surface receptors and activate intracellular signaling pathways that may normally be switched off during normal tissue homeostasis. Importantly, it doesn't just act on cancer cells, and we often see significant reprogramming of what we call stromal cells, examples being fibroblasts, either recruited or local within the tissue. Immune cells especially. There's a lot of work going on at the moment looking at how, for example, collagen receptors or other matrix receptors on immune cells might alter either their activation or exhaustion. And I believe you have an immunotherapy session coming up later in this course. We know that it can both directly and indirectly alter cancer cell sensitivity to therapies, and I'll touch on some of our work on that toward the end of this lecture. Importantly, it can also act as a physical barrier to the delivery of our therapies. So this capsule, or this sort of fibrotic wall almost that you can see that encapsulates tumours, can actually prevent chemotherapy from being able to enter into the tumours. I also mentioned that we see increases in stiffness, we see changes in intratumoral pressure, which can cause blood vessel collapse as a result of matrix remodeling, and all of these can act to physically stop chemotherapy from entering into the tumours. And the other thing, which has not been touched on yet, is that it can provide a physical highway for cancer cells to spread or metastasize to other parts of the body. And so these dense and thick collagen fibers don't always orient in this way that you can see here. This looks like a kind of a circumferential organization. In some tissues such as breast cancer, we actually see radially orientated matrix fibers, which known as tumor-associated collagen signatures, or TACs, if you're interested in looking that up. These are known to also be indicative of poor outcome and metastasis in patients. And so Not only does it provide, as I say, biochemical cues, but it can also provide physical routes of dissemination. Now, just want to touch briefly on really what's seen as the key architects of the tumour microenvironment in terms of the extracellular matrix, and that would be the cancer-associated fibroblasts. These are the ones that significantly produce large amounts of extracellular matrix. Cancer cells, I will say, cancer cells do produce extracellular matrix. However, they tend to be in a lower overall quantity. That said, they do also tend to secrete, let's say, more exotic or unusual matrix components, those that you typically wouldn't see within the tumour. And that's important because a lot of these cancer-secreted matrix molecules actually act as scaffolding molecules to change the organization of other matrix molecules. And so this sort of combination of large amounts as well as changed organization is really what underpins the importance of the matrix. Now, as I briefly alluded to earlier, this sort of matrix remodeling occurs both at primary tumour and secondary sites and is driven by a number of factors, typically secreted by cancer cells, but also in some cases immune cells. And this, as I mentioned before, feeds back to dynamic reciprocity. There it is again, that term, which really just sums up this intracellular, extracellular communication and co-regulation. And so really the premise of a lot of these approaches is that if we can target that deposition or remodeling of the tumour matrix, can we find a way to break that cycle and ultimately improve treatment and patient outcome? So what I want to do over the next sort of maybe 10 minutes or so is just talk about how we study some the extracellular matrix in some different tumours and then talk about the role that it might play. So over the years, there's been a lot of tissue clearing protocols being produced. This slide is a little bit out of date now. There are certainly a lot more than just those ones shown up here. And essentially, these have all been designed to enable you to image whole organs without the need to section in 3D. In fact, I've seen recent ones where whole animals are now cleared and made transparent. And this has been fantastic. It allows us to image very deep into tissues and really map cells within that three-dimensional context. However, most of these approaches, in fact, almost all of them, are designed to improve imaging of the cells and were not designed to image the extracellular matrix. In fact, many of them actually involve partial digestion steps where they degrade the matrix to allow tissues to swell in order to help improve resolution. And so a couple of years ago now, while I was still in Copenhagen doing my postdoc, a couple of postdocs got together and said, well, there must be a better way to do this for extracellular matrix. And so we came up with a way to decellularize tissue. So we remove the cells using a vascular shunting approach. And so these images here are really just representative of the different surgical procedures that you can use to use animal vasculature to essentially decellularize tissues from the inside out. And so what's more important is showing you these images. And so what you can see, this is over time, is that the shunting of vascular tissue, decellularization agents through the lung tissue, which is what we're looking at here, leads to that clearing of or removal of cells. And so on the right-hand side here, this is after 18 hours, this is decellularized lung tissue. And so this paper was published in 2017 for anybody who does want to go and look a little bit more into it. But I'll talk you through some of the parts of it in the next few slides. Importantly, we showed that our decellularization approach doesn't affect matrix integrity, but is really good at improving imaging, proteomic cataloging of the matrix in that three-dimensional structure and spatial organization. And so here are some examples of this here. These are dark field microscopy images, just for those of you who aren't familiar. So there's no fancy microscopy here as such. But what you're looking at is tissues. So this is from our breast cancer models, and on the right-hand side, our pancreatic cancer models. And you'll see that we've got various tissues, liver, lung, and hopefully you can appreciate is that the structure of these tissues is still fully intact. Many of them are still clearly identifiable, but there are no cells in any of these images. So this is the lobe of the lung, part of the liver, again, liver over here. In fact, you can see the fenestrations of the liver within these tissues here. And so this just really emphasizes just how much matrix you have in tissues and how important that's going to be in controlling and regulating cell behavior. Here's just another example. So this is the pancreas. This is a spleen of one of our mouse models. They can be stained for your favorite ECM molecule. This is just an example of collagen 4, and you hopefully can see this sort of grape-like or ductal structure of the pancreas here on the right in green. And this can be imaged in whole mount, so we don't need to section and process these. The other thing is, because it's whole mount and there's no need to section, is that we can look at the extracellular matrix organization over scales of magnitude. And so this image itself is a tiled image of the mammary gland, stained for collagen 4, which is an important basement membrane protein, and SHG, which is collagen 1, some of the same as the images I showed in some of the earlier slides. The scale on here is 500 microns. So these images are about 3 millimeters by about 5 millimeters, which is, for those who are familiar with microscopy, huge field of view. And what hopefully you can see is that this is normal tissue. This sort of, again, grape-like ductal structure. These are the mammary gland, or the terminal end buds, where all your milk gets produced, supported by a collagen 1 sort of fibrous matrix. And so what we can do is we can begin to zoom in into some of these regions. And so what we can do is zoom right in. And so what you're looking at here, this is collagen 4 still. These little grape-like structures are, as I said, the milk ducts. But what you'll see, these sort of vessels that are snaking over the outside, these are the capillaries that sit on the outside of those mammary ducts. And so you can see that once we've removed all the cells, even deep within tissue, we can begin to map that three-dimensional organization. Now, we can do this in our tumor models quite nicely, and so that's one of the key ways we actually begin to understand what's happening in the tumor microenvironment and how it's important. And you can see, so this is a decellularized lung, so there's no cells here present again. This has got developing lung metastases in it. You can see it really doesn't take an expert to see that there's a region up here in the top left with a big white arrow pointing at it, and also down here in the sort of center, where the matrix looks very, very, very different. It's very dense and opaque in this image. And so we can begin to zoom in and we can begin to look at this. This is just SHG, so collagen 1, looking at how the extracellular matrix within that tumor microenvironment appears in relation to that matched next to it, right adjacent to that developing lung met. And if you remember those images from one of the first slides I showed you, when I talked about the differences, this is exactly where they came from a 3D decellularized lung metastasis. But that's not all, because one of the key things we're interested in is understanding not just what happens inside a tumor. And so this is collagen 4 staining within a developing lung metastasis. You can see this sort of very fragmented organization. And also what happens in the normal lung. So we can see this is the normal alveolar structure of the lung stained for collagen 4. But what we like to call transition zones, that region where tumor essentially meets host. And you should hopefully be able to see that there's a very big difference as you move from inside a metastasis through this transition zone. The collagen 4 changes organization, becomes very dense, very disordered, and moves out into normal lung tissue. What you'll also see is the beginning to see, hopefully, is this white dense collagen matrix being deposited. And if you remember back to one of the earlier images, showing that you often get this encapsulation. And this is important because obviously the tumor microenvironment here, the matrix will give the tumor cells inside the metastasis one set of instructions. But as they move through this, either at the primary tumor or in metastatic sites, into these transition zones, you're likely to be encountering a completely different set of extrinsic cues. And if they break free and or move and migrate into normal tissue, again, that may be a third set. And so we're interested in understanding how do these different regions, especially at things such as these invasive edges, feed back into progression. We also like to make lots of pretty pictures as well. And so this is just an example of how we're creating three-dimensional reconstructions of multiple matrix components. And so what you'll begin to appreciate with just 4 ECM components shown on this right-hand image is just how dense that extracellular matrix is. Now, we don't just do this in mouse models. We actually got a program in the lab where we're doing this in human tissues, working with a number of different collaborators, and where we can take tissue. This is a breast cancer example, take tissue post surgery. This is just an example of normal tissue shown here in blue. You can see these are this is the ductal structure of the mammary tissue. The purple is the collagen 1. Again, inside the tumor of that matched patient, you'll see that there's a significant loss, for example, of collagen 4 organization. So again, that matrix has been heavily remodeled within the tumor. And we're also doing this actually in pancreatic cancer models, and we launched the Australian Pancreatic Cancer Matrix Atlas a few years ago now, where we're actually collecting patient samples from those undergoing surgery. And again, just look at this schematic in the middle, looking at the importance of how the ECM changes between the sort of marginal or invasive edges of the tumors versus the core tumor region, and also further afield. And so those are just an example of how we're also able to apply many of these imaging techniques to patient material as well as to animal models. The other thing we can do, which is nice, which is it really allows us to study the interaction deep inside tissues, is recellularization. So once we've removed the cells, we can then introduce back in cells. And this is just a very quick example showing that we can reseed tumor cells back into either tissues such as the lung or back into decellularized tumors, and begin to ask how they interact with the matrix and image in real time. And that's ongoing work, actually, not by a number of labs around the world are now using these approaches to really unpick what it is the matrix is doing to the cells. One of the last things I want to show you in terms of the imaging is that we can also do imaging of the matrix in real time, intravitally, so inside live and living mice. And this is work that's been pioneered by a number of individuals. Paul Timpson here at the Garvan, Adrian Hawkins down obviously at WeHi, which many of you may know. And up here at Garvan, what they use is these titanium window imagings, which looks like a small washing machine window, I guess, which is essentially put into the abdomen of mice and allows you to essentially image the organs underneath. And this can be done for weeks on end, and it allows you to go back to the same spot in either an organ or a tissue or a developing tumor and look at what might be happening. And the mice are perfectly fine with this. Hopefully you'll see there's this video running. There's a little window on the mouse. This is courtesy of Paul Timpson's lab, and you'll see that this allows you to pop this imaging window on a microscope and can re-image many times. And what that allows us to do is to look at how the matrix, so this purple again is that collagen 1 matrix, how that might change over time as a tumor grows or in response to a particular therapy. And if you combine that with fluorescently labeled tumor cells or immune cells or whatever cell you're interested in, you'll be able to actually image those interactions in real time as well. So one of the other things that we like to do, and this actually has been facilitated significantly by our decellularization technologies, is to catalog that matrix. And so one of the important things we do is proteomics-based mass spectrometry analysis. And I'm not going to spend too much time on that, but what I want to just say is that what it allows us to do by removing the cells is really make in-depth analysis of how that matrix is changing within healthy tissues versus tumours, metastatic sites versus primary tumours. And that then gives us a number of leads that we can then begin to dissect mechanistically as to the role that they might be playing. One of the things that we obviously don't get when we use proteomics is that we sort of blend everything up into a bit of a smoothie, for want of a better word, and then catalogue what's in there. What we lose is that spatial information. And so recently we've been working on mass spectrometry imaging as a new way to study the matrix in cancer. For those of you who aren't familiar, mass spec imaging is essentially capable of imaging a number of different small molecules, for example, proteins, lipids, metabolites, but it maintains that spatial information. And so very briefly, essentially, it involves putting a section onto a special slide, blasting it with a laser to ionize the various analytes that you're interested in, and then running mass spectrometry at each of those individual coordinates. This creates essentially a series of mass spectrums across your tissue, and then allows you to create maps of the distribution of those specific analytes as you move across the tumour or any tissue of interest. Now, what we've been doing recently is to develop this, and we recently published a paper on a new technique called HitMap, which allows us to begin mapping tumor microenvironment matrix within, so this is a H&E of a breast cancer. You'll see this H&E in the bottom left-hand side of the slide here. It's fairly homogeneous. It's pretty much all purple. But once we begin to overlay the different matrix spatial maps, you can see that they have very, very marked heterogeneity. And we can begin to look at how they, particular ECM components might relate to one another and how they overlap. Don't forget that matrix, individual components never operate in isolation. They're always part of larger supramolecular structures. And so we're often interested in finding out what else is sort of co-regulated with particular matrix components. What we're also doing is generating segmentation patterns. And so, again, this is just another breast tumor. This is a H&E. You'll see maybe you might be able to identify a little bit of histology within this if you've got a good eye. But by looking at how matrix signatures specifically cluster within the tumor, we can begin, and so that's what this center image is showing you. This is 5 clusters where matrix signatures most closely match one another, and so you can begin to look at how heterogeneity within a seemingly otherwise homogeneous tumor might be important. And actually, we believe that a lot of this microenvironmental heterogeneity underlies and feeds into a lot of the cellular heterogeneity that we're seeing come out at the moment. And so one of the most sort of, exciting things that we're doing or trying to do at the moment, and this is working actually very closely with Alex Swarbrick here at the Garvan, is to integrate our spatial transcriptomics programs where we can spatially map microenvironmental changes back to transcriptomic programs and look at how cells are changing transcriptional programs. And that's actually quite important because this allows us to really ask the question of, well, what happens in the microenvironment and how is that sensed and responded to within the cells present within that region, be it cancer cells, immune cells, or other. So what I want to do really now is sort of try and run through why is the matrix important and really highlight some of the work that we've been doing. And so I've probably alluded to already that patients often don't respond to current therapies, and that's mainly because tumor cells are very good at finding ways to escape, but that includes the generation of these protective niches and the creation of tumor microenvironments that really blunt the efficacy of our therapy. And so we now accept that it's really going to be more powerful to target multiple elements of that tumor microenvironment, either together or sequentially. And so by essentially taking away or removing those support systems of the tumor, such as the matrix, we may actually be able to augment efficacy of already approved standard of care therapy. The other thing I just want to highlight is that we can use matrix signatures to help stage cancer. So just as we've seen large amounts of molecular phenotyping coming out, we also argue that you can now, or we should also use matrix stratification approaches to complement that. And this is just an example we're showing here. Purple on this is that collagen 1 again. We can see we can very easily have patients with high, medium and low levels of collagen 1 within those tissues. And actually, given the longevity of a lot of matrix components, most of the ECM components in your body last for days, weeks, maybe even months. In fact, some of the collagens in your body you're born with and you will die with. So this is sort of a historical record of what's happened during tumor development. And so if we can really tease out both the biological and mechanistic sort of understanding of what this is doing, it'll allow us to begin to sort of subtype certain tumors. The other thing I will just say is that in almost every tumor biopsy, there is extracellular matrix. In fact, in some tumor biopsies, there's only matrix and no cells, and historically pathologists have been unable to score tumors if there aren't any cells. And so we know that there are millions of archived bits of tissue out there that we can go back to. And so not only can we image, we're now developing spatial proteomics approaches that we can apply to tissue microarrays as well. Okay, so how are we doing for time? What I want to do in the last 10 minutes or so is just talk about a research example of some of the work that we published a couple of years ago now, looking at that cancer cell matrix feedback in pancreatic cancer. And so this, like I said, was mentioned, it's published in 2019 in Nature Communications. For those of you who find this interesting, I would suggest you go and have a look over the paper. Really, I'm going to give you the sort of the highlights and the punchline, but there's an enormous amount of work that really advise you to check out the paper. And so this work started around the idea of trying to understand the role that p53 played in pancreatic cancer. And so we have two genetically engineered mouse models. So we all know p53 is one of the major oncogenes, but there are two types of p53 aberration. One is a loss of p53, and our mouse model is a flox model, which is represented here in blue by the KPFLC, and this is loss of p53. The other is a gain of function mutation in p53. So they both have the same KRAS mutation. They have different p53 mutations or loss, and everything else, in theory, remains identical between the two models. What we're interested in is that these loss of p53 models are highly tumorigenic, but they're very poorly metastatic, whereas the p53 gain of function are highly tumorigenic, but also highly metastatic. And so most people attribute this to the p53, that either loss of function or gain of function. But what we noticed when just looking at the tumors was that in the loss of p53 model, we have a much lower stromal or matrix component in that tumor microenvironment than we do in the gain of function, as you can see here in these picroseries red images. And so we reasoned that, well, maybe there's something in the extracellular matrix that's feeding back along with that p53 difference that might be important. And so to test this, what we did is we took mice from both these models. We isolated their tumours, and we took the cancer-associated fibroblasts, the CAFs, the known sort of architects of matrix remodeling in tumours, and some matched KPC cancer cells. So we have cancer cells and cancer-associated fibroblasts from each model. What we then did, and this is kind of a long story short, is a number of proteomic characterizations of exactly what it is that these KPC CAFs are secreting. What is it that's pumping out into the microenvironment or not, and that might be in some way playing a role. And so what we did, so this is just shown here with this heat map. This is sort of highlighting a number of differences between what we know as called as the matrosome here being the proteome of the matrix. And one of the ones that struck our eye was essentially this protein that we saw called heparan sulfate proteoglycan 2, or perlecan for short. And this is very highly conserved protein. It's incredibly important in binding to not only growth factors, but other matrix molecules and scaffolding that extracellular matrix. And so it controls organization, assembly, integrity, and cell interaction. And so this is secreted in very, very high levels in the p53 gain of function tumors compared to the loss. So again, cutting to really the punchline, we did a number of experiments to manipulate this perlecan within those CAFs, and then looking at co-implantation studies and how that might affect the microenvironment. And one of the take-home messages is that depletion of perlecan in the CAFs only leads to changes in local invasion at the primary tumor. And so this is the fox and the mutant model just shown here, but knockout of perlecan in CAFs only. And so this actually made us realize that, well, actually just by knocking out one matrix component from the CAFs in these tumors was enough to begin affecting invasion. And it also leads to a significant decrease in metastasis to the liver. Now, what I didn't say on the previous slide, which I just want to iterate here, is that actually the CAFs will have started their life as normal pancreatic fibroblasts or pancreatic stellate cells. So the only difference between the CAFs is that the tumor cells that grow and develop within the pancreas have a different p53 mutation or loss. The CAFs don't have this. So what's happened is that the cancer cells have re-educated in some way the local fibroblasts during the onset of that tumor to create either mutant p53 or loss of p53 educated CAFs. Basically, what this is then arguing is that that education process and then the subsequent secretion of the high levels of perlecan might be a strong or a key factor in controlling the metastatic potential of the p53 mutant cancer cells. The other thing we're interested in doing was to just check at how depleting perlecan might decrease or might affect chemotherapy. So this is just the time to metastasis graph from the previous slide and the overall changes in survival. So this is just knocking out perlecan in the cancer-associated fibroblasts. But we found that by adding chemotherapy to our knockdown compared to the wild type, so we're looking at purple is the cancer cells co-implanted with normal wild type CAFs. If we knock out, so if we add chemotherapy, so purple to green, we get this small improvement in survival. In the context of a knockout of perlecan in the tumor microenvironment, chemotherapy appears to be even better and extends survival. And I'm not going to go into this, but you can certainly read the paper. We showed that this was through the presence of perlecan delays the response to gemcitabine Abraxane, which is the standard of care chemo, and allows cancer cells to essentially escape the genotoxic effects. So just to sum that up, as I said, this is a very large paper, is that p53 mutant cancer cells, and I didn't talk about this, but through intracellular pathways regarding NF-kappa B signaling, secrete high levels of TNF-alpha, which is a known activator of fibroblasts, to educate CAFs within that local environment, and we call these mutant-educated CAFs, coming from the p53 mutant cancer cell tumours. This triggers these CAFs to secrete large levels of perlecan into the extracellular space, into that tumour microenvironment, and that presence of perlecan is enough to generate both a pro-invasive and chemoprotective niche. Therefore, blocking perlecan, and so we showed this with genetic manipulation, and we're now following this up with domain blocking antibodies, is likely to lead to the loss of this generation of pro-invasive niche and chemoprotective niche. So just to start summarising everything and bring it all to a close, what we know that is as tumours grow, if left untreated, we see a large deposition and remodelling of the extracellular matrix, recruitment of activated stromal cells or CAFs, and this leads to progression and ultimately dissemination. Now, a few years ago, actually a couple of papers out of Raghu Kaluri and Ben Stanger's lab in the US actually showed that if you, I'm arguing the tumour matrix is bad, so they put forward a couple of papers that showed that if you completely remove the tumour matrix, that tumour stroma, so you've completely changed the tumour microenvironment, and this is through blocking the ability of the fibroblasts to essentially create this matrix. What happened was that the tumours grew faster and spread wildly around the body. So that actually suggested that, well, hey, whoa, hang on a minute. Maybe there are some elements of that matrix that are restraining the tumour and that we can't just completely ablate it. Incidentally, what was really interesting was that if you did do this, then immunotherapy, immune cell access into the tumours was significantly enhanced, and that it's likely that that would increase the efficacy of immunotherapy. So the argument now, or at least the thinking in the field, is that if we can normalise or re-engineer the matrix rather than completely ablate and remove it, so return that tumour microenvironment back to a more normal looking microenvironment in combination with tumour targeting therapies, then we will likely be able to remove those pro-tumorigenic support systems and ultimately improve efficacy and outcome. So last slide, I promise, just to summarise all of that, the matrix is everywhere. It is all around us, even now in this very room. I say that as a joke, but I also say it seriously. You cannot think about and study cells in isolation in any disease you work in. You should always be thinking about the three-dimensional context in which cells exist in, and in particular, that scaffold, that matrix, which really acts as a nexus of communication between all cells present within tissues, healthy or diseased. Hopefully I've convinced you that homeostasis and remodeling are really important in not just normal tissue homeostasis, but also in tumours, and that they are an instrumental part of the tumour microenvironment. I've shown you some cool imaging technologies that we're using now to try and study the importance of the matrix and how it changes in diseases such as cancer. And also that there's a huge amount of unexplored targets, biomarkers, prognostic indicators that we're only just beginning to tap into. And this can involve repurposing old drugs, for example, antifibrotic compounds, or generating new drugs, or even targeting downstream effectors of cell matrix interactions, blocking the ability of cancer cells, CAFs, to see and respond to pro-tumorigenic matrix cues. As I mentioned, we might be able to start using matrix signatures to further stratify patients who might benefit from additional stromal targeting therapies, and that by normalizing that matrix, we can hopefully improve the efficacy of current standard of care therapies. So with that, I think I'll finish, and I would love to take some questions.
Thanks very much, Thomas. That was such an interesting and informative talk. I know I've never really considered the ECM components as therapeutic targets, and so I'll definitely be reading that Nature Comms paper after this. That was cool to hear about. And so we'll take questions from the audience. Please put your hands up and ask your question here, or pop your questions in the chat and I'll read them out. So I know we have students who have volunteered to ask questions today as well. And while we're doing the Q&A, I'll just remind everyone to respond to the poll Kerry has shared in the chat as well about your preferred format for WeHi training workshops. And while we're waiting for questions, I'm happy to kick us off. I was thinking about your using the matrix stratification as a method to stage cancers. I thought that was very interesting. Is there a correlation between matrix structure and patient outcomes? Can you use that ECM structure of patient biopsies as a prognostic tool as well?
Yeah, that's a really great question, and I sort of alluded to it a little bit when I was talking about the tax signature, which is from Pat Keeley, was the original proponent of this. What they'd shown was at the invasive edge of tumours, you get this radial alignment, so fibres that sort of extend away from a primary tumour, and that correlates. So they generated like a tax 1, 2, and 3, and it's a little bit subjective, but it actually tax 3 highly correlates with poor survival, tax 1 good survival. So that's an example of the architecture itself, so just that physical alignment. But what we're actually beginning to realize is that sometimes just loads of a particular matrix isn't necessarily the best indicator, but the ratiometric amounts. So if you get an increase in one or decrease in another, can have, again, that similar effect, or be correlated closely with survival. And it comes down to the fact that they all interact with each other and they scaffold one another and change their, the organization. And so that's actually a new way that we're thinking about it, is rather than, well, that's just high and that's just low and that correlates, is to say, well, how do these nuances, how do these things all come together? And, especially in that scaffolding sense, an increase in one with a concomitant decrease in another may be more powerful than just looking at one alone. And that speaks to architecture, although we don't quite necessarily understand how the architecture is being changed yet.
That's really interesting, thanks. I'll take the question from Harry, who's got his hand up.
Hi Thomas, thanks for the talk. You were speaking about how kind of in the developmental stage, the ECM can be quite similar, even between different tumour types. I was wondering, based on that, as well as CAFs, if there are any other sort of infiltrating cells that aren't usually in the ECM that are common across various cancer types.
That's a really great question. So we tend to sort of lump them, I suppose. We have, fibroblasts and CAFs, and I suppose in different tumours you'd have the precursors to those. So stellate cells is a good example in the liver or in the pancreas. And then we obviously lump most immune cells together when we talk about immune infiltration. At the moment, I can't say that we've looked into whether there's additional cell types. I mean, there will be, obviously, all of those cells that are normally present. A good example would be, I suppose in your case here, within pancreatic tumours, do things like islet cells play a role? Are they somehow involved in that tumour microenvironment and remodelling? My simple question, I don't know. We haven't looked whether there are cells outside of that. And it's a great point. We should really consider everything rather than just those that we're familiar with.
I'll go to the chat now. Sophie has asked, is it possible to upregulate parts of the ECM which promote tumour containment while also targeting the ECM to allow immune cell entry?
Yeah, I think that would be the million-dollar question. So we've got some programs of work where we are essentially doing the first part. So we know that some matrix components are tumor suppressive. They seem to at least, I guess, restrain or constrain tumors. Some great early work by the likes of Mina Bissell and co showed, and this was many, many years ago now, several decades ago, was that if you take transformed mammary epithelial cells and put them into normal matrix, then they actually almost behave like normal cells, suggesting that actually a normal matrix can, for a while, just restrain that sort of malignant phenotype. And more recently, there's been some great work coming out from some colleagues over in the US, where they've actually been looking at aged versus young matrix. And with aging, we see a lot of changes happen in the matrix. The most common one that we're probably all familiar with is we get wrinkles as we get older, as the balance of matrix molecules changes. And so aged ECM appears to have effects on promoting cancer, whereas young ECM seems to restrain. And so there are definitely work underway to say, well, what is it that restrains, and can we push that to essentially be upregulated? Of course, the question is, how is it controlled, and how can we upregulate that? I'm not sure we're at that process where we can sort of, you know, inject in matrix components yet to attempt to restrain it. But no, it's a great point. That's definitely one of them. Just speaking to the second part, whether you could upregulate one, downregulate another that might be pro-tumorigenic, I mean, that's really where you want to start going, removing or pruning away those that you know might be pro-tumorigenic, whilst retaining those anti-tumorigenic functions. But that's a great point. It's certainly work that's ongoing all around the world. And I would say probably in the next 10 years or so, we'll start to see things like that coming through.
That's really cool, thank you. I saw Anna had her hand up. Anna, if you've still got a question, please pop it in the chat or put your hand up. Hanadi asks, how does extracellular matrix remodeling relate to tumour dormancy?
Right, brilliant question. There's been some super exciting work coming out of the labs of people like Cyrus Gujar over in Seattle at the Fred Hutch, that are basically looking at how single cells interact with different matrix components, especially within perivascular niches, within the brain, within the bone, and so on. And so what we find is that at that single cell level, your cells are often in touch or in direct contact with various elements, and that they have that, as we were just talking about in the previous question, that kind of tumor suppressive effect. Now, a matrix component is not going to kill a cancer cell, but it may stop it from overtly proliferating and colonizing. And so that's where the dormancy angle comes in. And it speaks to what is known in the field, and I didn't mention, but something known as the seed and soil hypothesis that was originally proposed by Stephen Paget many, many years ago now. And that's this idea that a tumor cell requires that correct microenvironment in order to colonize. So the seed requires the right soil. And so this is critically important in dissemination to secondary organs, where a tumor cell finds itself in a very new or it's never experienced before environment, and the elements of that restrain it. Over time, something happens and it escapes from that, and then obviously dormancy, the dormant cells become reactivated. But yeah, there's some great work. Cyrus Gujar is a great person to look up, and I know that and he's been written a few really good reviews on it for those who are interested.
Cool, thank you. Anna, if you wanted to ask your question.
Can you hear me?
Yeah.
Oh, sorry. Thanks. Some of the questions have been asked. That's why I put my hand down, but I'll just still ask. So what about the polarization of the collagen? Like, does it have any effects in the development of the microenvironment, or that you can do anything with that?
In terms of the imaging that I showed?
Yeah.
Yeah, so the imaging which of the collagen, the polarized light imaging, well, that's it's basically exploiting the anisotropic properties of the dye. So when it binds to the dye, we can essentially measure. It's a surrogate for how many dye molecules are present and how tightly they're packed to one another. And so it actually gives us really useful information about bundling, density, thickness. And so that actually, by extracting different elements of that, can tell us about the tumor. It's obviously a static sort of measure, but it gives us more information than just a H&E that says, oh, there's matrix. And so yeah, we do extract some of those different bits of information. It's a little bit more bulk, so obviously that's done on your sort of standard imaging, a standard light microscope. We tend to use the second harmonic generation imaging, that SHG imaging that I showed, all these beautiful purple and white strands. That allows us to really get down to almost that single matrix fiber level, and then start asking about the architecture and the role that it might play. Does that answer the question?
Yeah, so far you don't have much info to know.
No.
Okay, cool. And just a very quick question, just out of curiosity, you know that in your PDAC tumor model, the overexpression of perlecan, how exactly that creates the chemoprotective niche? Like, is it like a physical barrier, or it's some modulation of receptors, or how does that?
Yeah, absolutely. I mean, you notice I totally skipped over that bit, and so read the paper because it'll definitely there's so much there. But what I will say is that we actually carried out a number of FRET biosensor readings of cell cycle, so using CDK1 FRET biosensors. And what we see is that we get an altered cell cycle in the presence of perlecan. And what that does is that therefore, so all of your chemotherapies essentially disrupt or try to disrupt DNA, causing cell cycle stalling and so on. What we see is that due to sort of a slightly delayed or altered cell cycle is that you get a delayed response to your chemotherapy. And so we didn't necessarily track back to know which receptors were being activated that was leading to the change in cell cycle, but we know that that was likely to explain. So the chemotherapy partially works, but it has a blunted effect. And so the problem with pancreatic cancer is it's hyper-aggressive. So any slight blunting in your efficacy is quickly going to be overwhelmed. So that's why, but there's some beautiful cell cycle analysis in there that gives you an insight into really how that's affecting the proliferation.
Super. I'll read that. Thank you so much, Tom.
We had a question from Antoine, who said your decellularization of tissue technique is incredible. It says, I was wondering if you had ever tried to stain for cell membrane markers anyway to see if fixation might cross-link some membrane glycoproteins with the ECM. This way, maybe even with decellularization, you might get an idea of what cells are around.
Yeah, so that's a great point, and that's, I probably didn't make it entirely clear. There's no fixation of the tissues, so they are still fresh. We never stain for cell membrane, primarily because when we did we were looking for cells, and so we tried a number of things. We were looking for phalloidin staining to see if there was any cells, DNA content, and so on. However, it is a great point in one sense because receptors and fragmented cell membrane may still be present. We haven't looked at it specifically. The decell approach does use detergents, so I do wonder if it might be washing out most of the sort of lipid-based things such as cell membranes. But no, we've never looked, and it's a really interesting question.
Cool, thank you. Krish Neal asked if the decellularization and reseeding process, he said it was a really interesting as a way to study ECM remodeling due to cancer, and asks, are these protocols also being used to study tissue transplants?
Yes, and there's been some amazing work by people, and this is partly where some of the tips and tricks that we got have come from. And I've actually recently been talking with a chap down in Melbourne actually about who's got a very similar approach. And so this idea of recellularization, recreating matrix in order to create humanized material. So there's a lot of work trying to take, for example, pig tissues, decellularize and remove those, and then recellularize with human tissues. It's not quite made it into the clinic yet, but it's definitely going that way. Mainly because I know it's getting better, but trying to 3D print an entire matrix structure is still pretty challenging. We're still a way away from that. That would be the ultimate goal. So yeah, it's tissue engineering and biomedical engineering sort of approaches is where a lot of this has come from. We're almost in a way using some of our hard work to apply this now to what happens in that diseased setting when things go sideways, for example, in tumours.
Right, thank you. We've got a couple more questions in the chat. I wasn't sure, Gemma, do we run a bit over? Is that okay?
If it's okay with Thomas.
Sure, yeah.
Yeah, there's a couple more coming. Yeah.
Thomas.
Right.
So Raymond asked, he said, thanks for the great talk, and asked, do you think chemokines binding to HSPG play a role in driving/inhibiting metastasis or immune cell infiltration?
Yeah, you again, I said it's a large multi-domain protein. It has a number of growth factor binding domains on it. Nothing gets past your students. They're awesome. Yeah, absolutely. We didn't look into that role, but we know that there are a lot of growth factor binding domains as well as matrix scaffolding domains. So that's what's really happening now, is that we're actually doing some work following up with the AstraZeneca antibody alliance to specifically generate antibodies targeting domains. And so the idea is that we can block different functional domains, be it a matrix binding, a growth factor binding, a cell receptor binding, and then start to tease out what's the most crucial, what's critical. Is it the fact it binds other matrix proteins and organizes that structure? Or is it just sequestering a growth factor that's known to trigger invasion? I suspect that it's probably going to be a combination of all of them, as many of these things are, and that it'll take us a long time to tease out which is the most important.
Thank you. Jackson says, thanks for a great talk, Thomas. My question is in regards to transplantation animal studies. To get some tumours to grow on mice, sometimes matrigel and growth factors are required to allow for tumour initiation. How does this affect extrapolating data for determining driver mutations in the cancer, thinking in the context of your KP and KPC experiments?
Yep. So you're right, there are some models that do require that. It's possibly down to this idea that you need some kind of supportive niche just to give them that foothold. In the paper I discussed, we avoid where we can the co-implantation with matrigel. Some of the models do require, especially in grafting into the pancreas. The reason matrigel is used in the pancreas is because it has a nice tendency to gel when it gets to about body temperature. So it's a liquid on ice. Anyone who's worked with it knows you have to keep it on ice, otherwise it starts to go nice and thick. And upon implantation into, for example, the pancreas, which is a very diffuse tissue, it gels, and so that actually holds the tumor cells in place whilst they engraft. Ideally, we would love to move away from that. But what's important is that in our models is that we have, for example, the Perlecan, we had Perlecan knocked out calves with cancer cells, Perlecan wild type calves with cancer cells. We then have the Phlox model. Each of those is obviously co-implanted in the same way. So any effect that matrigel is having would be applied to all settings. What I thought he was going to ask a question about, well, if I just have a minute more, was that can we use our repopulated to reimplant back in as a way to look at the effects that might have. And that's something we're actually really excited about doing at the moment. So we can say, well, we've got a bit of D-cell tissue that we can put cancer cells back into. Let's reimplant that within that 3D structure and then look at what happens. Does that affect metastasis, response to therapy, and so on? So that's some of the other cool work, which maybe I'll be able to talk about in the future at some point.
That sounds very exciting. Thanks. Our last question is from Jeffrey, who asked, the images of the ECM give impression of it being almost cobweb-like, mostly open and not occupying much of the volume, even though images were decellularized. Presumably the ECM contains water and is a lot less viscous than intracellular matrix/cytoplasm. Is that right?
Mm-hmm. Yeah, all correct. The one thing I will say is that the beautiful imaging I was showing you was one of hundreds of extracellular matrix components, and so there was that one slide we'd stained for 4, and it looked like a much more dense. So when we look at individual components, there does appear to be quite a bit of space. However, when you start to layer in, I mean, at any time there can be 400 or 500 matrix components present in different amounts within a particular tissue, and so that certainly begins to, similar to the cytoplasm, begins to start crowding. In terms of viscosity, matrices are often very well hydrated. That hydration can change, obviously, depending on it's controlled really by the level of proteoglycans. We don't see, unless it's a specialized tissue, like the vitreous humor in the eye, for example, we don't see a sort of viscous property in the sense that we might with a very high protein soluble protein-rich fluid, because a lot of extracellular matrix is insoluble. So it tends to be deposited in insoluble matrix. The only difference, I guess, would also be the blood, which has a soluble matrix. So I would say that your cells are definitely, as a viscous fluid, probably more viscous, but that there are a lot more than you can definitely see in the imaging I've shown you present within there. So
great, thank you so much, Thomas. I think that was the last of the questions today. Awesome, really appreciate your time. That was fascinating stuff. Thank you.
Thanks for the invite.
Yeah, thanks, Thomas. That was great, and a huge amount of interest from the students.